Thanks for the kind introduction. Hello to everyone again. Today I will be sharing with you some of the recent trends and advancements in genomic research. Most of the examples are from the latest literature. Apologies in advance if you get a bit information overload by the end of this 1 hour lectures. First, I will describe briefly about my career background, followed by a general introduction on genomics before we look at some of the most popular applications of genomics. Then I will describe the top 10 trends in the field of genomics based on my personal opinions and the current literature before we conclude on the future prospect of genomics. If time permits, I will spend a little time introducing some of the exciting research projects from my group in plant functional genomics. During my undergraduate bachelor degree in biology at Sheffield University in the UK, I gained a solid foundation in diverse topics of biology. Then I continued pursue in plant molecular biology, working with the model plant arabidopsis on the role of plant cell wall protein in leaf development during my PhD. With exposure to biomechanics and mathematical modeling, after doctorate graduation, I started my position as a lecturer and started establishing my own research group in plant functional genomics with specialization in transcriptomics. More on this if we have time near the end of this lecture. UKM, the National University of Malaysia, is a comprehensive public university with many greeneries. It is 50 years old this year and among the top 150 universities in the world according to the QS ranking. So let's begin our lecture today with some brief history of genomics. Can you guess what are all these pictures represent in the key milestone of genomics? First, we have the Mendel genetics study on P's trait inheritance, which was rediscovered in the 1900s. Then we have the structural heritable material, DNA, described by Watson and Crick based on X-ray diffractions by Rosalind Franklin. The first commercial DNA sequencer by Applied Biosystems in 1986, which was invented by Frederick Sanger in 1977, which allowed the beginning of the Human Genome Project in 1990s and completed in 2003. The draft was published in 2001. Then we have the first synthetic mycoplasma bacterial genome by J. Craig Venter in 2010. What's after 2010? Can you guess? Yes, some of you get it right. The CRISPR-Cas genome editing in 2013, which allowed precision genetic engineering. So what's next? You might become one of the inventors for the upcoming breakthrough. Okay, what is genomics? It is the study of whole genome of organisms, whereas genetics mainly refer to single genes and heredity. There are two main aspects, the structural and functional genomics. Nowadays, computational genomics is also growing fast. That itself can consider a field, but in this talk, I'm going to just focus on these two aspects. Structural genomics deals with the physical nature of whole genomes. This includes generating the reference genome, which is often haploid, meaning half of the total genetic makeup, since majority organisms are diploid. For example, in the human, there are 22 pairs somatic chromosome and 2 sex chromosomes. These involve mapping, sequencing, annotations, and of the genes. Then we have the physical map, which allows the genomic structure as seen in the cytogenetic map, with features of hetero and euchromatin representing the dark and light bands, respectively. Nowadays, we can even map out the 3D location of nuclear genome using specialized sequencing technique, which will be described later. Lastly, structural genomics also include comparative genome analysis to identify synteny, which is the conserved relationship in the gene order, as can be seen in the figure of the mouse chromosome segment conserved in human chromosome. This is the mouse, this is the human. And this study of genetic variation is key to evolutionary biology. Another definition of structural genomics is a genome-based approach with high throughput methods using X-ray, NMR, or cryo-electron microscopy of structure determination by a combination of experimental and modeling approaches. To put it simply, it is a field to describe type 3-dimensional structure of every protein encoded by a given genome. But we mainly focus on the first aspect. As for functional genomics, it covers the expression of gene usage, such as the study of all gene expression known as transcriptomics, or the whole of proteins in the cell or organism known as proteomics, and their regulations, according to the central dogma of molecular biology. So the DNA code for RNA, which is processed into mRNA and then translated into protein. So at this different level, there lies the complexity of genomic regulation, in which one gene can code for multiple transcripts, which can be translated to different protein isoform with different post-translational modifications. As a whole, genomics actually covers diverse fields of research, from fundamental comparative genomics to applied translational genomics, especially for clinical application. We'll touch on some of these in the later slides. So we will look at some of these applications. First, we have the disease genomic. We study the molecular mechanism of the cause of diseases. Then we have the pharmacogenomic in drug discovery. Genomic medicine deal with the diagnostic and therapy, which genome editing allows functional analysis through in vitro or in vivo testing in cell culture animal model. Lastly, synthetic genomic encompasses biodesign for genome engineering of novel pathway or compound through synthetic biology approaches. Some of the molecular technique, bioinformatics, and clinical application of genomics in molecular medicine is illustrated in this diagram. You can see the technique used, whole genome sequencing, host pathogen genomic, and transcriptomics were used to study all these different aspects of molecular medicine, and then this can be translated to vaccine development, drug discovery, and treatment guideline. So for disease genomic, which cover human, animal, and plant diseases, mainly focus on studying the mechanism and gene disease associations through sequence-based assay, and to characterize variant function usually curated in database. This is a well-known human disease gene association database, this GeneNet, with ultimate aim to enhance knowledge and biology of diseases. Another example for plant is the plant disease resistant genes database, where one can explore the pathogen receptor genes from diverse species of plant. As for pharmacogenomics, uses the genetic information to tailor pharmacotherapeutic decision for an individual. For example, the blood thinner warfarin depends on two genetic variants, the cytochrome C9 and the vitamin K epoxide reductase complex subunit 1, with a big range of dosage from 2 to over 6 mg per day, so depending on the patient genotypes. So furthermore, pharmacogenomics also aim to identify new targets for medicine and even drug repurposing. Genomic medicine uses the genomic information to guide and tailor clinical decisions for personalized medicine. In the past, the diagnosis of cancer relied heavily on pathology. Nowadays, molecular markers from the protein can be used instead to access patients' samples diagnosis. Some well-known examples include to assess the disease risk, clinical diagnosis, treatment choice, disease prognosis, and risk factor. Some of the examples include the Gale model for breast cancer risk assessment and the Framingham study models. So this is usually associated with genome-wide association study, the GWAS. This helps to improve patient cohort selection for clinical trial and also inform treatment strategy to optimize therapeutic benefit for patients. Some of the trends in genomic diagnosis and treatment include the expanded definition of self, then just your own genome, but also microbiome, and etc. And earlier diagnosis to molecular testing. As for treatment, which can be more tailored to target specific disease marker with improved response and drug safety. Even the incorporation of dosing optimization using mobile app technology is possible. Genome editing technology, despite being a buzzword only recently, since the advent of CRISPR-Cas in 2013, its development has come a long way since the study of various nucleases like ZF zinc finger nuclease and then the tail nuclease. It's interesting to learn a bit on the timeline of CRISPR from its first discovery in 1978 to its success as a gene editing tool in 2012. So this is a story to be continued. So what is CRISPR-Cas? It stands for Cluster Regularly Interspaced Short Palindromic Repeat. Basically, it's a sequence in the DNA, which guide a DNA cutting enzyme called Cas9 to specific sequence of DNA. Bacteria uses this system to recognize and destroy foreign DNA, such as attacking viruses, and we can use this to enable precision genome editing or for genetic engineering. By creating a customizable library of single guide RNA, we can be built to target different genes. Another technology known as CRISPR, the isomer interference, can be used to block targeted gene expression. It is the same principle that it just binds and recognizes the complementary strand. This is how the structure looks like with the single guide RNA and target DNA in actions. An example of genome editing in pluripotent stem cells create different cell lines for drug discovery. Cas9 mediate the double-stranded breaks. They are subsequently repaired by two mechanisms, either the error-prone non-homologous end joining or the homology-directed repair. So individually, they introduce different things. For example, this introduce indel insertion deletions, whereas this you can introduce another segment with homologous end to be integrated into the chromosome, and this can be used as a approach to create precisely edited clones. And this is part of the drug discovery toolbox enabled by the CRISPR-Cas technology. So it's a wonderful tool for functional analysis of various genetic elements by precisely targeting different elements of gene regulation, such as the promoter, and then the exon sequences, and even the 3' untranslated region through various techniques derived from CRISPR. Another promising area of genome editing is crop improvement. From traditional breeding to natural transgenic to man-made transgenic, the field has come a long way. Now plant molecular biology can target gene editing responsible for certain desired traits. As such, and there are more to come. For example, resistance against diseases and environmental factor, or crop with higher yield or better nutrition without allergen or to control weeds. Why genomic analysis is important in this context is because it's important to confirm the event of editing. There is a possibility of off-target editing, hence, most studies will include the off-target analysis as part of the pipeline. So nowadays, off-target analysis can be conducted on the Cas9 digestion site, be performed using long-read sequencing approach like the PacBio and also the Oxford nanopore sequencing technology without the need for gene amplification. Aside CRISPR-Cas system, other genome editing approaches, gene-based editing and also prime editing has been introduced more recently in 2017 and 2019 with better specificity, so less off-target perhaps. Then that's why it's more promising. So one can anticipate a truly precise genome editing in the future. However, we are still concerned about off-target genetic alteration. Aside that, the most common Cas9 is derived from Streptococcus pyogenes Cas9. There are also other variants derived from Prevotella and Francisella, known as the CPF1. This is a smaller and simpler RNA-guided DNA endonuclease than Cas9. So this can be used for various applications such as knockout, knock-in, base editing, prime editing, and etc. Apart from genome editing, it can be also used another type of system, another Cas known as Cas13a and Cas12 can be used as a detection tool, especially Cas13 that can actually degrade RNA instead of DNA. This has been used as a COVID-19 diagnostic test strip, which can detect even 10 copies of viral DNA, viral RNA also in per microliter sample. Lastly, synthetic genomics enable redesigning of organisms for truly genome engineering as compared to genetic engineering, which deal with small stretches of genomic DNA. Long stretches of synthetic DNA can be constructed. Like this is the timeline on the development of synthetic genomics. The one I mentioned is the 2010, the first one ever with the synthetic genome. And recently in 2019, we have the Caulobacter intensis 2, which is created by computer-assisted de novo chemically synthesized genome with minimal genome, and we can envision artificial cell comprising synthetic genome for specialized functions such as bioproduction of certain drugs, antibody, or compound in the future. Now, next part of this lecture will focus on the 10 current trends in genomic research. I'll briefly go through them to provide an overview of where the genomic field is heading. Don't worry if you don't understand some of them. The important thing is to be aware about these different topics, and then you can pursue further if interested. First, metagenomics constitute a cultivation-independent analysis of the collective genome of microbe within a given environment using sequence and function-based approaches. Basically, it's a genomic profiling of environmental sample, which also include your gut. Metagenomics can be considered the convergence between the field of microbiology and genomics with the advent of sequencing technology. So from the first coin of the cell or microscopy until now using sequencing base. So there are two types of shotgun metagenomics: targeted approach focusing on micro identifications based on amplicon sequencing of the RNA or ITS sequences for the species identification, whereas the non-targeted approach just sequence everything to study all the genes for inferring microbial community functions. Metagenomics, like any other genomics, rely heavily on bioinformatic analysis to make sense of the data through classification, clustering, and also the visualization to look at the functions and also the compositions of the microbial community. An important field of metagenomics is of clinical sample with many applications. I can see here, such as for the infection disease diagnostic, for microbiome analysis, and even include transcriptomics to look at the gene expression, and also can be used for oncology applications. Since metagenomic approach is culture independent, this method allowed the identification of many previously unculturable microbes. Gut microbiome has since become one of the hot research topics. Recently, there's a catalog of over 200,000 reference genomes from the human gut microbiome was published as a reference for the study of gut microbiome. For those interested, can refer to this nice review article. Second, the field of evolutionary genomics. The recent phylogenetic network analysis of SARS-CoV-2 genome is a good example of how this can be applied to infer the spread of COVID-19 on the different variants, A being the ancestry variant. So why we sequence the viral genome? Basically to identify the origin of the virus for population surveillance, to identify the transmission route, to track mutation and evolution rates, and also design and to evaluate the diagnostic tests. Currently, many vaccines are already in animal and clinical trial thanks to the genomic advancement. So this is like some of the timeline of one of the vaccine. So hopefully we'll find a long-lasting vaccine soon. So for genomic-based biosurveillance, there are currently quite a few of large global projects such as BioScan, EBP, and Global Virome Project, which hopefully can intercept pandemic before they occur. Some affiliated project network of Earth Biogenome Project are shown here. You can see that it involve many different types of projects. Third, the phylogenomics, as compared to phylogenetics, is the use of whole genome sequence to reconstruct evolutionary relationship between organisms. Shown here is a phylogenomic of over 10,000 genomes, which reveal the evolutionary proximity between archaea and bacteria in the domain level relationship compared to previous phylogenetic analysis using core genes or ribosomal protein. Furthermore, the phylogenomic can also be constructed based on synteny, which I mentioned earlier, the conserved genomic region on the order. So microsynteny refer to the relative gene order. So it's found that the genome organization of the mammal is more conserved than in plant because of whole genome duplications. Hence, there are more copies of the same genes in the plant than the animal. Thirdly is the population's genomic. Studies have gone beyond an individual nowadays to study the whole or different population. It relies heavily on Bayesian probabilistic statistics to infer gene selection to understand the selection forces acting on the different genes. So the evolutionary process of the genome, and a good example is the study of genetic selection in the malaria carrier mosquito, Anopheles gambiae, and now this field is moving toward global level population genomics. Furthermore, population resequencing can also be applied to investigate the demographic history of domestication, the genetic architecture underlying domestication trait, and patterns of selection. Here shown here is an example recently published in June in PNAS on the bitter gourd to identify some of the genetic architecture of signature of bitter gourd food traits based on the different variety, even including the Indian variety. So you can see this is the gene population structure based on the genome resequencing. Population resequencing is important to capture genetic diversity in crops which are not present in the reference genome. So one reference genome is not sufficient to cover all the different variety. Hence, with the resequencing to cover the variety, genome editing can be targeted. This leads on to the pangenomic. When multiple genomes are available for a single species, a pangenome can be constructed in comparison to the core genome, which excludes the accessory and the unique gene. So the pangenome includes from different individuals the genome diversity, so making a pangenome that covers all the diversity. So this allows to link more phenotype interest to their causal variant for understanding genetic instruction. A very good example is in tomato using Oxford nanopore long read sequencing of 100 tomato accession. This established the pan-structural variant as feature of structural variants. Many structural variants in the cis regulatory element, which is not inside a gene but the promoter, the enhancer region of the genes, actually responsible for the flavor, fruit size, and productivity of the tomato in the branching and fruiting pattern. So this is won't be able to detect if it's based on a single genome only. GWAS, as mentioned previously, is another important field of genomics, especially for genomic medicine. We study involve many patients to determine the risk associated with certain genetic variants, such as the BRCA genes in the breast and ovarian cancer. The first GWAS is on the age-related macular degeneration in 2005, which is 15 years ago. It's revolutionary for the identification of genetic risk factor. The field is currently adopting more whole genome sequencing approach instead of using the more cost-effective SNP SNP array to determine the variant trait association. Based on the recent review of over 3,600 GWAS study over 3,500 traits, the trend shows an increasing sample size with more diverse ancestry, so from different population, and the number is keep increasing every year. And this is some of the well-known example, well-cited GWAS study. So, however, current GWAS are still dominated by the European population, over half, and this mainly focus on EG to measure phenotypes, and it is foreseeable that future direction is moving towards a more inclusive GWAS for less common diseases. Like this is what is still yet to be done, so the future is moving towards this direction. On the other hand, meta-analysis of GWAS provide additional statistical power in identifying weaker gene disease association. You can see the scale of over 100,000. The minimum is like, in this example, is like over 200,000. So GWAS can also be used to identify trait of interest in the crop. So this example in tomato. So GWAS is not just for human. Single-cell genomics is another fast-growing field of research. Individual cells can now be isolated for analysis independently to profile cellular diversity. And this example, the sequencing of single B cell is considered a single B cell. So individual cell was captured in the fluidic system from the 10x genomic, and this allowed the sequencing of individual cells to discover the antigen reactive antibody based on the heavy and light chain sequences. In plant, there are also many applications of single-cell genomics with diverse applications based on different techniques. I'm not going into details on this. Just give you one example. You can actually sequence individual pollen mother cell or even the megaspore mother cell to identify the crossover pattern between chromosomes during meiosis. So when the gamete cell is produced, you can use single-cell genomics to study that aspect. Recently, a single-cell transcriptome atlas characterizing the aging tissue in the mouse has been published just 2 weeks ago. This provides a comprehensive resource for the cell biology community that offers detailed molecular and cell type specific portrait of aging. It also provides an essential companion to the genome. The genome provides only a blueprint, but it does not explain how the gene are expressed in a cell type specific manner or how the gene expression changes over time. That's why we need this transcriptomic atlas in doing so. It can be said that single-cell genomics will keep developing with all these techniques, with increasing number of single-cell genomics study using different techniques and toward more dynamic and integrative studies. So number 7 is cancer genomics. ONCO stands for cancer, which has gone global with the involvement of many countries. It's a large-scale study. So there are many objectives for oncogenomics, such as molecular and histological classification, defining the driver landscape, to study the resistance mechanism and understand the genomic basis for metastasis, also mutational processes and repair deficiency. With the Cancer Genome Atlas, now it's moving towards a pan-cancer atlas from many tumors to the individuals, with the integration of multi-omics cancer study and towards a larger scale global and populational study. Number 8, the structural genomics characterize the genome structure by mapping the three-dimensional chromosome architecture, which involve multi-scale exploration. So you can see this is the deepest at the DNA, but if you go out, it's like at a larger macro level. You can see that it can be at the compartment or the topologically associated domain, or it can be up to the chromatin loop. So you go down deeper is the nucleosome cluster, and then you get the DNA. So it has been studied at a different level for exploration. For different level, it involves different technology based on The field is gaining momentum. On the discovery that genome structure alteration can contribute to disease treatment. For example, here comparing the healthy cell and cancer cell at the 3 different levels, at the compartment level, or the topological associating domain, and the chromatin. So you can study at different scale. And currently, it's also an alternative target for anti-cancer therapy based on the 3D genome. With all the different omics data, provide an ideal approach for integration, such as the case of cancer study. So these two not represent the two proteins interacting with each other or relate to each other. Even genomic medicine is fast moving towards multi-omic for a more holistic understanding. Another example is the multi-omics of study of twin initiated by NASA to study the effect of living in the International Space Station for 1 year. You can see that many data are collected. This diagram summarizes the different level of approaches for the multi-omic integration. So they are element-based, pathway-based, or metagenomics-based. So you can refer to this review. Lastly, it's inevitable for the application of artificial intelligence in genomic research as the data volume is ever increasing. Machine learning, deep learning, and the natural language processing are very useful in mining and processing all the data in scientific literature. Example of AI and machine learning application include extracting the genomic and epigenomic variant for clinical utility, to identify genes, to predict genomic association, to predict protein function, or predict the sequence and specificity of DNA, RNA binding protein. This is an example of artificial intelligence application in constructing an evolution pathway of SARS-CoV-2. To summarize, genomic research are largely driven by genomic medicine. More data will be generated by the third generation sequencing. Large-scale global population genomic, pan-genomic, and single-cell genomic will become more common through research consortia. GWAS will continue for the identification of polymorphism associated with risk for its complex hereditary traits such as cancer, CVS, and diabetes, and is moving towards meta-analysis. Multi-omic system integration of biological big data and analysis provide a more holistic framework. Automated analysis pipeline and also AI-driven technology and analytical tool will further accelerate the genomic research. So what the future hold for genomics? The current trend is pointing towards the need for a new generation of researcher with strong biological background and computing skill as a data scientist to tap into the wealth of genomic data such as AI-assisted disease prediction shown here. So we need a new group of scientists like you or the future generations. The global genomic market is on an uptrend, which worth over 30 billion by 2027, with the compound annual growth rate of 7.7%. So the future is bright. Because of the this opportunity is largely driven by application in clinical genomics with the workflow simplification and the decreasing cost of sequencing. This is accelerated by automated analysis pipeline and software tool for many different applications. This is just to support the involvement of artificial intelligence in the genomic market. There is an error in the figure. If you notice, it's pointing here the largest region as the North America, but it's the Asian region. I think that this should be the truth. The upcoming market should be in Asia, with China and India being the highest market with highest potential. So the future is bright in our regions. In perspective, genomic data is increasing exponentially. Hence, there's a bottleneck in the storage and the retrieval. And the trend is now going toward cloud computing like Amazon Web Services. The bottleneck is on the functional validation, data creation, and analysis for meaningful application. Hence, this leads to the wet lab automation, artificial intelligence, and machine learning. More and more people will be obtaining their own whole genome sequence, so this allows precision medicine to be a reality in the future. Genome with epigenome and microbiome will allow the integrative healthcare. With an expanding global market in the field of genomics, we need more computational expertise and experimental biologists to contribute to genomics from a new generation of biologists. The data security, data sharing, and data ownership issue will become essential factor to be considered in the light of universal human rights. Hence, the blockchain technology will be useful for this data encryption. The advent of molecular medicine undoubtedly drive a dilemma in ethics. So we need to relook at the bioethics for bioethics 2.0. Lastly, for the latest genomic breakthrough, you can refer to this great source of literatures. That's the end of the genomic aspect. Since we have some time left, I'm going to introduce about the research at the Institute of System Biology. It's the first Institute of System Biology in Malaysia, also in Southeast Asia. We was established 15 years ago on 24th March, and we seek holistic understanding of biological complexity by providing a dynamic infrastructure and research environment all in one place. In system biology, we integrate genomics, transcriptomics, proteomics, metabolomics, and bioinformatics, as well as glycomics to review the biology of plants and microbes. We focus on crop improvement and molecular exploration of tropical plants and microbes. Shown here is an example of using rice for crop improvement. We use multi-omic approach assisted by bioinformatic analysis, in vitro, and transgenic technology. And this leads to synthetic biology and product commercializations. This is a general research framework we use for biological knowledge discovery using omics. And we also target some genes functional analysis, and all combined integrated for the reconstruction of secondary metabolite biosynthesis pathway, which allow metabolic pathway engineering. To illustrate this, we use the Flagship project on polygonal miners, commonly known as Genome in Malaysia. It's called Vietnamese coriander in general, and the synonym is Persicaria minor. It's well known in Malaysia because of the pungent smell and the taste used in the laksa dish. It's also used traditionally for stomachache and also dandruff, to treat dandruff. So we applied transcriptomic approach in the past, the express sequence tag library, and later on we use the next generation sequencing for the small RNA degradome and transcriptome analysis. We also perform proteomic analysis with protein purification and characterization and also some enzyme protein structure analysis using X-ray crystallography. We have done a lot of metabolite profiling on the flavonoid, terpenoid using GCMS and also LC12MS. And all these are combined for the reconstruction of secondary metabolite pathway through data curation in database, also development of bioinformatics tools such as PRIM and NGS Frontier. We also developed the polynerous oil as an essential oil for fragrance that's like with health benefit. And currently there's a lot of bioactive compound isolations ongoing. The known bioactivity include antioxidant, antimicrobial, anti-cancer, and cytotoxic activity. And we are now developing it as a preventive supplement for neurodegenerative disease such as Alzheimer's. And we are also trying to metabolite engineering in the yeast Saccharomyces cerevisiae. My group have performed transcriptomic and metabolic profiling of methyl jasmonate, which is an elicitor, induced polygonal leaf. So we perform a time course study using 150 micromolar of methyl jasmonate, and then we profile the volatile organic compound based on GCMS, and we perform the transcriptome differentially expressed gene analysis using total and small RNA. Then we validate using the reverse transcription quantitative PCR. So we demonstrate that high amount of aldehyde and terpenoid are induced on day 1, which later on reduce over the days of 3 and 5. These inductions correlate with the increased expressions of the enzyme related to the synthesis of terpenoid and also the terpene biosynthesis pathway in general. So there's a correlation. And based on this understanding, we use the genes to metabolite engineer the yeast and bacteria for the production of some useful flavonoid based on pathway reconstructions. And this is the work still ongoing. Because my specialization is transcriptome, I will just introduce some of the transcriptomic works at INBIOSIS using system approach. As I mentioned, we focus on the discovery of new genes and biomolecule, and also for crop improvement. These are some of the plants that we work on, including the Jatropha for the biodiesel. Then we have the Kratom for the mitragenin production. We have Gaharu, the resin, which is valuable for fragrance. This is a custom I mentioned. We have some seaweed, firefly, bagworm, which is a menace for the oil palm industry, Arabidopsis, grouper, papaya, and we've done a lot of work on Mangosteen, which I'm going to describe later, and my personal research interest on the carnivorous plant here. First, my master student work on the papaya endosperm culture, trying to identify the triploid plant, which is seedless. So we may get some triploid callus. And then we perform genome-wide transcriptome profiling of Carica, papaya, and Burgeonic callus. In summary, we transform and try to study transcriptome-wide effect of the suppression of these ethylated 1 genes in the callus. And we found that the upregulation of certain flavonoid biosynthesis will support the use of the callus for biofactory. Then, for biomolecular discovery, I work on the bioprospecting of Nepenthes pitcher plant for commercially useful enzyme and to understand the molecular effect of plant hybridizations. This is shown here also the tissue culture of the very nice ornamental Nepenthes. Nepenthes current is like mostly in the tropical regions, even in the Sri Lanka and also Madagascar, so mainly in the Southeast Asian regions. There are many type of carnivorous plant depend on the different type of trap, either very sticky or very slippery. Pitfall trap are mainly slippery trap, and Nepenthes is one of the passive carnivorous plant with a pitfall trap. So you can see some active trap like the Venus flytrap using snap trap, and then bladder suctions in the aquatic Uticularia, and then the fly paper we stick to the plant, especially the Drosera species. So there are quite some pitfall trap species like Sarracenia, Darlingtonia, Heliamophora, Cephalotus, Catopsis, and Brochinia. All these evolve independently of each other and in different continents. And Nepenthes is one of the most diverse pitcher plant in the tropical regions with very diverse shade of pitfall trap. In my study, I focus on these 3 species with very distinct diet. The Ampullaria growing on the ground, they can actually harvest a leaf litter from the forest floor, whereas the Raphisiana is a true carnivore, and their hybrid is also a carnivore. So we are comparing the parents and the hybrid. And nicely, we have a common garden experimental terrace where you can study the plant in situ on site. We have to monitor the plant for control experiment so that we can harvest at the right time. Just for the protein and the transcriptomic, just within 24 hours of picture opening, and for the metabolomic profiling, we harvest 1 week for the full mature picture. So you can see the picture when it's the cross section, there's a waxy zone for the trapping, and then you have the digestive zone with the fluid and enzyme secreted for the digestion and absorption of the nutrient from the insect's trap, or also the leaf litter. So you can see also for the 3 different species, the longevity of the picture also different. Ampullaria live the longest with over 8 months old, whereas Rafflesiana has a shorter lifespan of less than 3 months, and the hybrid have the intermediate lifespan. We use the same system biology research framework of applying metabolomic, transcriptomic, and proteomic to understand the molecular physiology and also for bioprospecting. And we perform proteomic informed by transcriptomic using the PacBio approach and also the LCMS protein profiling approach. For metabolomic, we use LCMS. We have an in-house LC-TOF-MS that we can use, and we do the extraction, and we standardize our harvest and extraction method, and we try to understand the compound. Summary, and these are our finding on the Carnivory mechanism. So basically, when the fly, the prey drop inside the digestive fluid, to attract the fly, we have the compound like terpene, alkaloid, flavonoid, some of which are volatile for the aromatic chemicals. Also the pigment on the colorful picture, and the nectar will attract the prey. When the insect go inside, there's some naphthaquinone like bombagin, deeper inside drosteron and 5-O-methyldrosteron. This will drown the prey, like suffocate it, or make the prey dizzy or drowsy, like chloroform to human. So they will be like drowsy inside and then stuck with the viscous fluid. When they want to climb out, like for the ants, they were prevented by the wax, which is slippery. Okay, a deeper look at the molecular component. So the prey, even before the prey fall in, there is some protein already pre-secreted. However, when there is a prey, there is also signal transduction to secrete more enzyme. Previously, there are 29 protein and 36 protein reported, but currently we have identified more than 180 protein and over 200 metabolites. So there's a fluid secretions and fluid activation. I forgot to mention the fluid is in the picture fluid is acidic, or like our gastric juice. And the prey provide all these components useful for the plant: polysaccharide, protein, nucleic acid, and lipid, phospholipid. And the plant actually secrete different type enzymes for these different categories, such as glucanase, chitinase, and then neprosin, nepenthes, and then RNase, endonuclease, and further digested to the basal component like amino acid, monosaccharide, and nucleosides. So lipid has some lipid transfer protein and also acidic phosphatase for the phosphate. And these are absorbed by the pitcher. So there's a transporter for nitrogen metabolism. And for defense, there's something like protein, peroxidases and catalase that produce reactive oxygen species, which kill the pathogen carried in by the prey and prevent them from competing for the nutrients. And there is also jasmonic acid, which is a stress hormone that potentially produces secondary metabolite for the stress to reduce the oxidative stress in the fluid, including some flavonoid and terpenoid. So as a whole, the Omics approach helps us to better understand the Carnivory mechanism. We also found that certain proteins are continuously secreted in the picture fluid by performing protein depletion experiment. So we empty out the picture on day 0 and filter out the protein and replace with the empty fluid. Then we sample again on the day 3 after sealing. Then we notice there are actually a lot more protein being secreted. Some, for example, are replenished, but some are actually not. So what's next? Then we aim to study. So we study the protein compositions and notice the changes in the different category. And this is the SDS page gel showing the protein fluid in the different samples. And as a whole, we found that the transfer white shift from the photosynthetic energy metabolism, that means these are reduced when the endogenous fluid protein depleted in the young picture. So this is the day 0 picture and this is the day 3 pictures, and this is what happened during the new depletion. Currently, my student is working on the applied side on the recombinant expression and characterization of commercially useful enzymes. We also constructed a database to curate all these omics data, known as Nepenthes base, and the plant omics database of all the different Nepenthes and even the Genome and also the Mangosteen, which I'm going to talk about after this. So we hopefully can provide a comprehensive biology research portal for everyone. What are the implications from this work is that we can understand further on the evolutionary history of digestive enzyme carnivorous plant for the bioprospecting of new compound enzyme for clinical and industrial application. And this understanding can help us to develop a bioreactor for plant protein, like a company that is trying to use the feature Nepenthes species as a bioreactor for the plant-made protein. Okay. For mangosteen, I think most of you will be familiar with this queen of fruit. They have a succulent and juicy pulp with a very the dark purple color pericarpal shell, which stain your fingernails, and they are actually high in xanthone, which is anti-cancer. We performed genomic and functional genomic study on mangosteen. And for the mangosteen genomic, we performed a genome survey by performing Illumina sequencing, also peptide sequencing. Currently, we are working on the plasmid and metagenome sequence assembly. What we found, we were actually comparing two different varieties. The common mangosteen, which is known as Mangis in Malaysia, is round shape, whereas the Mesta is more oblong shape. We noticed that the Mesta variety is actually bigger in genome size. Both of them are actually very high in heterozygosity, which is surprising because they are apomictic. That means they are produced without sexual reproduction, without fertilizations. You can see here the chromosome is complex. They are they are almost 74 to 110 numbers of chromosomes with over 6 picogram of diameter. That's like almost 6 gigabase. It's like human genomes, deployed of human genome. Okay. For the functional genomic side, we perform gene profiling type of study on the seed germination. I can see here there is actually no embryo in the mangosteen seed. It's not as recalcitrant because it cannot be stored for long. And Garcinia type is very special in that there is no embryo inside the seed. The plumule and the radicle is formed de novo during the seed germination step. So it looks like the tissue culture with organogenesis happen during the seed germination. So we perform time course analysis, identify the DG for between 0 and 3, 3 and 5, and 5 and 7 to check what gene is differentially expressed. And we perform cluster analysis on this gene expression across these 4 time points. And mostly you can see that there's this group, the upregulated and downregulated, they're antagonistic. So one group is downregulated while the other group is upregulated and vice versa. And also look at the metabolism pathway. You can see that there's anaerobic respirations will happen on day 3, and this coincides with the shift in the transcriptome. We also perform network analysis of mangosteen with using the network constructed from INBIOSIS. INBIOSIS is a model plant that we compare with. So the red color represents the gene involved in the germination, is upregulated, whereas the blue represents those genes upregulated during dormancy. So in comparison, we look at the changes of the day 0 across the 4 time point, we notice that between 3 and 5 is most resemble of the pattern in the dormancy germinations in INBIOSIS. So which means that day 3 to 5 is the key to the seed germination of mangosteen, and this is the key time point during the transcriptional reprogramming as well. As a whole, the transcriptomic profile allows us to have a detailed understanding of the molecular physiology of mangosteen seed germination by understanding what happened at different time points, and we can have a better understanding to help with the propagation of mangosteen. Okay. And aside from those studies, I also collaborated with other colleagues in the institute to work on different plants. For example, working on the drought and salt-resistant rice. My student Lewen, she was working on the microarray analysis of transgenic rice or expressing this Austin binding protein, which confer a better drought resistance. And we have Mahsusun Hazani working on the transcriptomic type analysis of fruit ripening. You can see the xanthone or the color purple is like changing during the different time point of fruit ripening. This still work in progress. And I also studied the largest flower in the world, Rafflesia. So this is in collaboration with Prof. Kilian, and we look at the bud development and also the flower senescence. So looking at the expression of various hormonal genes involved in the regulation of flowering processes. This is a cross section of the bud. So basically there's only one flower for the whole Rafflesia without any stem structure, and we can reconstruct the pathway connection and hormonal signal transduction. We also work on aquaculture fish like the grouper. Tiger grouper is very popular, especially during Chinese New Year. The Chinese love it. They're at the restaurant and it's highly valuable. This fish, the flesh is tasty. However, this fish is affected by fibrosis, so we are looking at the transcriptomic response of the fish towards vibrio infections. Another disease that we look at is the basal stem root disease in oil palm, affected by Ganoderma harmonensis, and the student had graduated and she performed the glycomic analysis, looking at the glycan between the pathogenic and non-pathogenic variant of this fungal. So that's all the work from my research group. These are some most of the people working on those projects that I mentioned. And that's all from me. Any question, or you can visit my website for the latest information. Thank you for your attention.